4.3 Osmosis. Osmosis is a specialized case of diffusion. Specifically, osmosis is the diffusion of water across a selectively permeable membrane. So water is going to diffuse down its own concentration gradient across a membrane, and that concentration gradient for water is created because there are different concentrations of non-diffusible solute. We'll look at a diagram in just a moment and explain a little bit more about what that non-diffusible solute means. A solution is a homogeneous mixture of several different molecules. If we look at a solution, normally the solvent is a liquid. The solvent is going to be the material that is there in the highest concentration. The solute is usually a solid, and that is going to be there in the lowest concentration. So for example, if we had a solution of sugar water, the solvent would be the water, the solute would be sugar. Now let's look at osmosis. In this diagram we have an apparatus called a thistle tube, and that thistle tube is inserted down inside a beaker. The opening of the thistle tube is covered with a differentially permeable membrane, so it's going to act much like our own plasma membrane. Now inside the thistle tube we have a solution that contains 10% sugar. So if it's 10% sugar, that would be 90% water. Remember, it has to add up to 100%. And then in the beaker we have a solution that contains 5% sugar and 95% water. Now if we look at the diagram right in the middle, you see the red line. That would be the differentially permeable membrane, and those sugar molecules are trapped on one side or the other of the membrane because they are too large to cross the membrane. So these would be non-diffusible solutes. They can't cross the membrane, they're trapped. Now remember, there are different concentrations of sugar. On one side we have 10% sugar, on the other side we have 5% sugar. So there are also going to be differences in the concentration of water. Up on the top, that represents the solution inside this thistle tube that has 90% water. The solution on the bottom, which represents the water in the beaker, that would have 95% water. So now we have a concentration gradient for water. It's created because of the concentration of the solute, but the solute cannot move. So the water will move. And so the net movement of water in this diagram is going to be from the beaker across the membrane and into the thistle tube. So if you look at the diagram on the right hand side, you can see that the fluid level inside that thistle tube has increased. That is because water is crossing the membrane by osmosis and raising the level of the solution in the thistle tube. Tonicity is the ability of a solution to cause osmotic change in a cell. We're going to look at three different possibilities here. The first one is an isotonic solution. Now remember, this is referring to a solution that could be surrounding a cell. In an isotonic solution, the prefix iso means same, and it has the same solute concentration in the solution as that is inside the cell. A hypertonic solution, hyper means high, so a hypertonic solution has a higher solute concentration than what is found inside the cell. And a hypotonic solution, hypo means lower, so a hypotonic solution has a lower solute concentration than that is inside the cell. Now recall what happened to the thistle tube whenever we had different concentration of solute. That created different concentrations for water, and that creates an opportunity where osmosis can occur. the diffusion of water across the membrane. So let's look at some examples. First some animal cells and then some plant cells. If we place an animal cell in an isotonic solution that has the same solute concentration in the solution as that is found inside the cell, and so there's going to be the same water concentration on both sides of the membrane as well. And so there will be no net movement of water. We can still have water coming in, water going out, but there will be no net change in movement of water. However, if we place a cell in a hypotonic solution, remember hypotonic, the solution has a lower solute concentration than what is inside the cell. If it has less solute, then it must have more water than that is found inside the cell. So if you place a cell in a hypotonic solution, the net movement of water is into the cell. And this cell will take on water, it will swell, it can eventually even burst, which is called lysis. If you look up in the top right-hand corner, this is a picture of a red blood cell which has been placed in a hypotonic solution. It has taken on water, swollen, and is about ready to burst. If we place a cell in a hypertonic solution, this is a solution that has a higher solute concentration than that's inside the cell. If it has more solute, it's going to have less water, and so the concentration gradient for water in this case would be higher on the inside, less water on the outside, and so the net movement of water will be out of the cell. And so a cell in a hypertonic solution will lose water, it will shrivel up, it will shrink. This is called crenation. Osmosis can occur in plants as well. If you place a plant cell in an isotonic solution, again, there's going to be no net movement of water. Just as we saw in the animal cell, concentrations are going to be equal. If we place a plant cell in a hypotonic solution, it will again take on water, so water is going to move into the cell. Osmosis draws water inside the cell, but because plants have a cell wall, this cell will not swell up and burst, but it does create increased pressure inside the cell. This is called turgor pressure. This is very important in plants. This is what allows plants to stand erect and oppose gravity, because they have this increased turgor pressure inside the cells. If we place a plant cell in a hypertonic solution, water is going to leave the cell. Osmosis will pull water out of the cell, and so the cell will shrivel up. Now again, we have the plasma membrane inside the cell wall, and so the entire cell wall is not going to shrink, but the plasma membrane draws back away from the cell wall. You can see the photograph here taken through the microscope over on the right-hand side, and you can see the outline of the cell wall, and the plasma membrane has shrunk back into the center. Now, what you can see there are the chloroplasts, the little green chloroplast, and that allows you to see the boundary of the plasma membrane where it's pulled back away from the cell wall. The term for this is plasmolysis.